Welcome back, Year 12s. So we're going to move on to a bit more of a complex topic, something you haven't learnt in the earlier years. So should be a good video. All right, let's get into it. So RNA and DNA. Obviously, RNA is single-stranded, okay, and it has uracil, nitrogenous base, instead of the thymine. And obviously, that's DNA with a double strand, and it has thymine instead, which pairs with adenine. We looked at different types of RNA, so ribosomal RNA. So rRNA found within the ribosome is a part of it, not all of it. The rest of it is protein. Okay, tRNA, which will help in the process of transcription. And it's that T-shaped RNA, okay, with the anticodon within it. Cool. And we looked at messenger RNA too. So mRNA, which is essentially just a split gene where it's a single-stranded piece of nucleic acid, where instead of showing the whole entire gene sequence, it only has the opposite of the gene sequence. Okay, and that's made by RNA polymerase. Cool. All right, so we're going to look at genes and codons specifically in today's class. Alrighty. So we moved on to the next dot point. So that's this one here, as well as a little bit of the first dot point too. Cool. So central dogma of molecular biology. Essentially, it was founded by this person, Francis Crick. All right. And really, it's that information can be passed on from nucleic acid to nucleic acid, and then it can be created into a protein. Okay. So as you can see here, pretty much the summary is DNA can be turned into RNA, and RNA can also be turned into DNA. Okay, but only RNA can be turned into a protein, and that is an irreversible process. Awesome. So we have gene structure here. Okay, so as you can see in the image, we have our T, A, and C, so our thymine, adenine, and cytosine, and those three together make up what we call a DNA triplet. So once that goes through transcription and processing, and it becomes messenger RNA, we then refer to those three together as a codon. Okay, generally they're used interchangeably, but really it's DNA triplet and RNA codon. Okay, but you'll hear it essentially called a codon from now on. All right, and they have their opposing bases, of which that will become, so the base pairs. So as you can see, T, the base pair is A. All right, so that's the opposite. So when the DNA gets turned into the RNA, it's going to copy it based on this part of the DNA. Okay. So that's the A. You have the A on this side, which, it's a U this time though. Why is that the case? Because RNA, remembering, has the U. DNA will have the T instead. Okay, and then the C, G. Simple. So that's your triplet, that's your codon. And that continues all the way through the gene. We have multiple triplets making multiple codons. Now, there's a few different terms that are used when we're talking about genes and codons, okay, and they're shown in this table here with their description and essentially an example. So if we say universal, pretty much means that anything that's living uses the same codon and amino acid sequences. Okay, so whether you are a frog, whether you're a human being, whether you're a eucalyptus tree, or whether you're a Staphylococcus pyogenes bacterium, if you have UA, that amino acid is going to be leucine. Okay, so it's as simple as that. It's universal. Unambiguous essentially means a similar thing, but in this case it just means that UUA can only code for leucine. So UUA does not code for tryptophan. UUA does not code for methionine. Okay, it will only ever code for leucine. But that doesn't mean other things don't code for leucine. Other codons, should I say. And that's what degenerate or redundant means. Okay, so redundant or degenerate just means that there can be multiple codons that code for one amino acid. All right, so one codon will never code for multiple amino acids, but multiple codons can code for one amino acid. So in this case, UUA means leucine. UG also means leucine is brought. CUU, CUC, CUA, CUG, etc. That all creates the amino acid leucine. And lastly, non-overlapping. This is less important. Pretty much they just never overlap. Okay, so they're always 3 at a time. Boom, boom, boom. And they're your triplets, which then in the end in messenger RNA will make your codons. Okay, so they're always adjacent. And yeah, this is really where if things were to change, so if you were to just take out this A and put in a C, that would completely change whatever the amino acid is that's being made. Okay, in this case, whatever it was, AUU would then be made into CUU, which, as we can see here, creates leucine. And that's what a mutation essentially is in the genetic code. All right, here we have a representation of a codon chart. So the first thing to note is in the top right, it's got codons or triplets. Why is this not a triplet chart? I'll give you a second to figure this one out. That's right. If it has a U in it, that means it's got to be messenger RNA. Okay, so it's not DNA. So in this case, it's a codon. It's not a triplet. So that's why it's not a triplet chart. It's a codon chart. All right, so we have the chart here. So you can see there's the first base, the second base, and the third base, and that's how you read it. So say you had CGC as your codon. You can look at C as your first base. Okay, so C's here. Well, it has to be within this box. I don't know why it's doing that. That doesn't look very nice. It's going to have to be within this box. All right, so C. Our second thing was G, so it's going to have to be within this box. Okay, so C, G, so you're looking here now. And the third base was C. C, G, C, so arginine is the codon. Okay, the other notable thing is the start and the stop codons seen here. So AUG, it codes for methionine. That's the start codon. That's going to be the start of any RNA gene. Okay, and if that was to be shown on the DNA, it would be the opposite of these, obviously. So it would be T, it would be U, sorry, it would be T, it would be A, and it would be C, TAC. Okay, if it's the stop codon, that's just the end of your RNA, so it finishes the gene. Okay, so that's UAA, UAG, and UGA. These 4 are important to know. This is a quick activity I would run with my students. So just find the different codons using the chart. Okay, so go back 10 seconds and use the chart. Okay, this next part is the gene structure. All right, so we're going to actually look at the genes now in eukaryotic organisms. So making sure that you don't mess this up with our prokaryotic organisms, because they do have different gene structures. Okay, and we're going to learn about that later when we learn about the trip operon. But for now, we can just worry about the gene structures in eukaryotes. So what do we have? We start off with this region where there's lots of T's and A's together. Okay, it has an interesting name called, well, we call it the promoter region, but it's also named the tartar box, which I don't really like. I just call it promoter. But there's a lot of T's and A's there. Okay. Cool. We have the 5' end, which is going to be the start of the gene. We have the 3' end, which is going to be the end of the gene. Okay, and we have these exons and introns in between, and then the termination sequence, or the last part, which we'll look at as well. Okay, so promoter, exons and introns, and then lastly the termination sequence. And that's in order from start, or the 5' end, to the end, the 3' end. So we have introns, all right, regions of non-coding DNA, and essentially during RNA processing, that gets cut out. Okay, so before it gets, before it gets to the ribosome to be transcribed, the messenger RNA needs to go through RNA processing. Okay, this process here, and these introns are cut out. All right, introns cut out. We also have exons, which are the coding DNA. So you really want those in the complete messenger RNA, or the mature messenger RNA. All right, they're going to be transcribed and translated into the final protein. So the best way to remember them is exons are expressed, introns are cut out. Okay, so exons are expressed. So you can see that here. Exon, intron, exon, intron. The introns will get cut out, and then the corresponding colors will go into that mature messenger RNA once it's gone through the RNA processing. All right, termination sequence. I always say that when I see this image. The termination sequence represents a sequence of DNA that signals for the end of transcription. So that's where you'll find those stop codons. All right, right at the end of the termination sequence. So that's fairly simple. And the last thing we're going to look at is just a complete eukaryotic gene. Okay, so we can see that here. There's a few things we left out. The terminator sequence, also known as the trailer region, which I prefer that term actually. We have the promoter region, which was mentioned. The leader region, which was not mentioned, but that also can be there. That'll be important later when we look at prokaryotic genes. And then your exons and introns in the blue and the red, respectively. Okay. And you also have RNA polymerase, which is running down the strand and transcribing the genes. Okay. Yeah. So thanks very much for watching. Hopefully that helped. We'll continue on in the next video. Thank you.